Okay, so we are right on time, and in fact, 4 minutes as usual delayed to start. So I welcome one and all for this week's Wednesday colloquium. And just for those who are tuning in either through YouTube or coming in here for the first time for the colloquium, so I welcome you all. The NSF colloquium series is pretty old, and for those who are new to this, this was originally started by Professor Homi Bhava, who was our founding director. And the entire series, the colloquium series, brings together people from physics, chemistry, and biology streams to listen to one person who would speak broadly about the entire kind of research area the person is working in. And it helps connect faculty members and students to different problems, okay, and within TIFR as well as with the speaker. So it's been a very successful series in training students and as well as putting collaborations on the forefront. And I am pleased to welcome Professor Arnab Mukhopadhyay from the National Institute of Immunology to come over here and deliver this colloquium. In fact, he has come, and a budding biologist along with him is there in the audience. So I welcome both of you, and I want to actually formally ask my colleague from the Department of Biological Sciences, Professor Vidita Vaidya, to introduce the speaker.
Welcome, Arnab. It's lovely to have you here. We've been intending to have you here for a colloquium for a while, so we're really happy that you were able to make it. It's my pleasure to introduce Arnab. His trainings, he started his undergrad at Presidency College in Kolkata, then went to DU where he trained as a plant biologist with Professor Tyagi, and then migrated from plant systems over to C. elegans, the worm model that is used by our colleague Sandhya in the department. And he is perhaps ideally placed to be called a physiologist in the full sense of the word, because he is interested in fundamental questions of lifespan, longevity, health span, how insulin signaling, metabolism talk to organismal level systems to influence whole organismal level behavior as well as longevity. And, I think we are in for a treat, because this is a wonderful model organism in which to ask these kinds of fundamental questions about biology. So I'm going to hand over to Arnab with that introduction.
Okay. So thank you. So I was told that we would, have a lively discussion with a lot of physicists and chemists and some biologists. So, I've tried to modulate my my talk in that way, and it needed a lot of soul searching. So, yeah, pardon me if I don't get you into this zone. So I come from this institute, National Institute of Immunology, and we study defense against dark arts, which is our way to fend off challenges in terms of pathogen, in terms of our own cells when they turn against us, as in cancer. And I am surrounded by some biologists, and mostly biologists, some chemists and physicists, but they are all turncoats. Okay, so they speak in the language of biology. So when Jyotishman invited me and told me that I have a special slide for that. Yeah, this is what my reaction was, that you have to talk to string theorists and astronomers and mathematicians and physics of all kind. Right, so it really got me, and that's when the nightmare started. So I called my brother. So he's a physicist, and he said, "Okay, fine. We have had a lot of discussions on biology and physics. Don't you remember?" I said, "Yeah, we always had a bottle of whiskey in between us." So that's not going to happen today. Okay. So now let us think how I think about all this stuff. As you saw, that I'm a diffused kind of biologist. I don't understand most of the stuff. I just yeah, I quoted you right out of that. I didn't ask your permission, but yeah. So just like I'm not really a trained physicist. I'm not trained biologist. I'm not even trained mathematician and all. So I kind of think a little bit differently. So what I thought is that, biology as a discipline is the most indisciplined of all life sciences, right? You physics, you have fantastic formulas, like you have, and fantastic people from you whom you can draw inspiration about how to think about science, and I do from this gentleman. And the formula, I have no clue. So similarly, if you look at the chemistry of life, look at the complicated, biosynthesis of adenosyl cobalamin. I'm going to talk about this a little bit. They're all beautiful and, elaborate, disciplined chemical reactions, right? So this is gibberellic acid, one of the most difficult thing to synthesize by human beings, easily synthesized by plants. And so they're all disciplined, right? And if you look at biology, this is what you get: an abstract, science which follows all the rules of chemistry and biology. And what we really don't understand is, where did this sink got lost, that we have so much variation, so much stochasticity in the system, and students laughing at me, struggling to see why, each experiment is not repeated if every, reaction is followed in toto from physics and chemistry. So at the end of the talk, I'm going to try to convince you that this colorful thing is how we perform biology. Okay, so this name is performance. I don't know why it is, but so let me turn Aristotle in his grave by saying that he probably did not understand the depth of his, this very interesting way of putting human nature on paper. That is, humans by nature are social animals, and that because he was restricted in some ways or the other. But he is one of the most celebrated and one of the wisest man dead. So what he probably meant by these social interactions were derived by human interactions within the species, right? So the humans, we interact all the time, right? Like now I'm interacting with you. We interact with our colleagues, with our friends, with family members, and many of this interaction, most of them are bonding or leads to happiness. And then there are interactions which are conflict generating, and we have so much conflict in the Levant, and people are really suffering. So we also are interacting with the domesticated animals, our farm animals, our pets, gives us a lot of happiness, right? So these are all interspecies interactions. So this is the title of my talk, that we interact not only within ourselves but with all other species. So we interact with plants for our food. We also interact with them, to draw solace, because we love to garden. We also interact with wild animals, and there also that there are good interactions and there are conflict with elephants, for example, with tigers when we go into their zone and try to take their habitat. So all these things, I think he missed, right? He only looked at human interaction. The biggest thing that he missed was the interaction with microbes, because 2000 years later, that's how long we took to to look down into the world of microbiology. And had it not been for this gentleman in around 1700s who invented the microscope, as all that it opened the life of opened up the biology of microbes. And from there, we started understanding how closely we interact with the small microbes in our environment. But when you think about our interaction with microbes, all you can think about is disease, right? So, for example, we just went through this pandemic, and that killed 7 million people. We know how these So this is okay. This is yes. So then I will just tell. So you are infected with helminths, with protozoans, right? Most of you have got dewormed when you were kids, or when you were even adults. You were infected with rotaviruses when you were young, and of course, that pandemic of HIV during the 80s. You have Mycobacterium, which is one of the most potent and most intelligent pathogen around us, right? And we are trying to eradicate Mycobacterium tuberculosis from India by 2030, right? That's a national mission. So we are always interacting with pathogens. These are microbes, and these are all conflicting interactions. They cause pain, they cause sufferings. But what I'm going to tell you about is some very interesting interactions which we do not really think about, is the interaction with beneficial organisms, beneficial microorganisms, and that is called the human microbe interaction, or the interaction that we have with the microbiota. So to give you a perspective of the depth of this interaction, let me give you some numbers. And I forgot to acknowledge that most of this thing I've just picked up to the internet, and they're quite colorful, and it caught my eyes. So I just thought about this for a while, and I thought that we should not be offended if people call us animals. Why? Because we are not really animals. We are actually more microbes than we are animals, and you see the numbers. So if you look at these numbers, this is the approximate number of genes that we have in our body. Our chromosomes can bear. Right? That's ballpark number, plus minus a couple of thousand. This is the number of non-redundant genes that the microbes that are living in our body, mostly in our digestive tract and urogenital tract, in the respiratory tract, that's the number of genes they are giving us, right? So a human being can have around a few trillion cells. The microbes give us additional 10 to 100 million cells, and these are residing in our body, right? They're beneficial mostly, and some of them, I'll come to that. So there are 10,000 different species of microbes residing in our body and our skin. And so if you now compare, say, our nuclear genome, our chromosomes, like for me and anybody of you, they're 99% similar. But now if I extract the gut bacteria from your body and my body, they'll be 80 to 90% different, depending on the food I eat in Delhi. And you probably, two of you will have similar, because you're eating out of the same canteen, same kind of food. So there will be some uniformity in your microbiota, but very different from mine. So this is an interaction that many of us did not really realize to that extent over very, very long time. And especially me, because there was no reason to look at that interaction unless you would have called me for this symposium. So let us go a little bit down into the biology. And I tell you that this is a very diverse population of microbes that infest us. Okay, so if you look at the organ system, not all of them have equal amount of microbes. There are a lot of discourse about whether all the organs are actually sterile or not. Right. So let's not go there. But if you look at the stomach, it has around 10 to 1,000 CFU per ml of that of microbes of different genera, species. And as you go down into the colon is infested with microbes. Okay, and most of them are beneficial. Okay, and this diversity of microbes are very important. We probably know a little bit about each microbe and their contribution. Most of them are correlative. There are fewer studies which are causal in nature, but we do know that the microbial diversity in this, in the digestive tract is of extreme importance. And why? Again, I reiterate that microbes gives us supplemental genome, things that we cannot synthesize in our body. The microbe helps us synthesize enzymes, small molecules, neurotransmitters, and as a result, they have a huge control over our physiology. Now, they also account for the missing heritability, right? So if you look at the Mendelian laws of inheritance, not everything can be explained with that. So microbes, on the other hand, in some cases, tells us that these additional genomes also put constraints on the theories of inheritance. It is a great source of energy. It can make short chain fatty acids, which our gut cells can take up easily and metabolize it. It enhances our immunity, and most importantly, it provides us with micronutrients which humans cannot make. Vitamin B12, vitamin K. So we depend a lot on these microorganisms. And so you can imagine if there is a dysbiosis, that could cause disease. Now, this is a more
the genetic material of the microbe and the human is not going to mix, right?
no, but the point is that the microbes make many other things which could affect phenotypes in one way or the other. Ultimately, you are looking at phenotypic.
Yeah, but there would be many factors which affect the phenotype, and I guess the microbes are one of them.
I didn't get you.
There would be many factors which affect the phenotype.
Yes,
this is one of them.
Yes, of course. So that's why I said that they explained some of the missing heritabilities in order to, because they are additionally producing stuff and affecting the genetics in a way that we don't understand. That's what I meant by those words. So this again summarizes a large number of things that the microbes do in our body. One of the things, it modifies the nervous system. It controls our behavior by synthesizing neurotransmitters in the gut. It's very interesting, something that I'm interested in. Even the pharmaceutical companies have to really worry about how their drugs that are administered are modified by these microbes and how they will end up in your systemic circulation, right? They have the power to modify detoxified drugs, correct? So I told you about vitamin biosynthesis. They help us in maintaining our gut epithelia, right? And they compete out pathogens. So the better health of your microbiota reflects in your innate immune response to pathogens. So they do a large number of good things. Now, we are doing things wrong. We are trying to scrub ourselves every day, shampoo and all that sprays and all. And this is a classic example. I love this example but this is n equal to 1 experiment. This is Mr. Haji. He lived in a very odd area in Iran, and he lived a ripe age of 94, took no bath for 60 years. And some of his people around him suddenly decided that he needs a bath, got him bathed, and he died shortly after that. This made big news. I don't know how many of you saw that on CNN, BBC, everywhere he was there. So, I mean, this is n is equal to 1, but this tells us that how important the microbes are, right? So you don't necessarily have to take bath. Don't quote me every day. I know this in the family, it runs like that. So, so now, see, we are doing something wrong because we are killing the diversity of the gut microbes that we have inside us. And the way you can kill your diversity is by changing diets. And by changing diet means that if you are not having diversity in your diet, the kind of food that you are eating, if you are having, processed food, if you are on antibiotics for a longer time, if you're living in an environment of stress, all these can dramatically affect your microbiota, right? And this is very well studied. Okay, so, and most interestingly, if you see this, if you eat something, it can affect microbiota just like that. And that's quite natural because your microbiota are microbes like E. coli. They only take 20 minutes to divide, right? So the effect is quite dramatic. And there are some studies, very interesting studies of Japanese, and this is long time ago, Japanese people who relocated to US, and the microbiome done to these people, 3 months later, the microbiota completely changes in dynamics, and because they were having Western food. So this tells us that the microbiota is a dynamic population of microbes, and to keep it healthy, you have to also invest in them. So, and the more diverse your, I mean, it's a very nice word I picked up here. The diversity matters on the plate and in the gut. So how do you maintain diversity in the gut? And that's why you see that there's a huge industry, a booming industry in the world, that is the prebiotic, postbiotic industry, which is $70 billion now. They're pegged to be 124 billion or something in 2035. And this is a great area to invest on scientifically, as well as if you have money, extra money, just put it in this. So what are prebiotics? Prebiotics by definition, So prebiotics by definition contains fibers and carbohydrates which are difficult to digest. So I'll come to this later, and you can see some green leafy vegetables and eat your vegetables. That's what we have always heard from our parents, right? This is why it matters. And there is another class of biotics which are called probiotics, which has the live organism. So you have probiotic milk, you have probiotic dahi, all kind of, so they are infused with good quality lactobacillus, right? So that they provide you more a trillion cells every day. There are many other variants of this. You have now postbiotics. People who are immunocompromised, who have gut barrier problems, cannot have probiotics because these bacteria can also infiltrate their gut barrier, right? So they are administered postbiotics that are derived from this bacterial population. Then there are phytobiotics, which are flavonoids, which are infused in your food. So this is a big industry trying to maintain a healthy gut bacterial population. So this tells you that scientifically there are dominating examples of how your microbiota can influence your health, and people are becoming more aware about this. Now let us switch gear and come to this business of aging research. Okay, so and I have to transition you from gut dysbiosis to aging. So I thought about many different ways. This is, I thought, was one of the better ways of doing it. So one of the things that happen during aging is gut dysbiosis. If you look at the population of microbes from childhood through say 100 years of 100 age, you'll see that the dynamics change all the time. The dynamics change when you transition from a toddler to an adult. The dynamics would change if you're obese rather than a healthy individual. If you are on antibiotics versus you are malnourished, everything seems to have a different signature of microbiota. And that makes a lot of sense, right? From the fact that I told you that what you eat will determine how long you live and how healthy is your microbiota. So again, reminding you, microbiota it has critical protective function. It has structural functions in terms of maintaining your gut barrier, your immunoglobulin levels, your immune system development, metabolic functions like preparing your body to produce vitamin B12, which you either get from food or your microbiota makes it for you. There is no way you do not have the biosynthetic pathway to make it. And it also digests the undigestible fibers that you are eating with your veggies, increases iron absorption, and a bunch of different things that we know of. So very interestingly, you see the link between gut dysbiosis and aging. Most of the diseases that are linked to gut dysbiosis are diseases of aging. And the examples are right there. Parkinson, Alzheimer's, many neurodegenerative diseases, cardiovascular disease, diabetes, insulin. These are diseases of old age. They were not supposed to happen at early age. But unfortunately, because of our dietary habits, because of the way we have changed our lifestyle, people are getting faster, aging faster in the population, right? So you see instances of age-related diseases as young as 20s and 30s. And what we know about is that much of it is food-derived. We'll talk about it a little bit more. But how much of it is actually driven by gut bacteria is something that we do not know. And this is a statistics, of course, from NIH. But this tells us that as you grow older, the burden of diseases increase phenomenally at a population level, right? So it is important that we understand the biology of aging. We understand how microbes, the beneficial as well as, of course, the pathogens, how they interact with human body. So at multiple levels, we have to understand how animals interact with the microbes and the vice versa. And you have to also imagine that they have all co-evolved with us, right? So we know these microbes. We have seen them. We have mechanisms already built into our body to avert big diseases. So the fundamental questions that I am trying to ask in my laboratory is how does microbiota affect aging? It's a part of the, trying to understand part of the things.
Few slides back, we were showing some statistics like at different age, what is the kind of microbes you have. Is it applicable for all kinds of population like in
No.
India or
No.
what is the
No.
statistics for
So the problem is you cannot, I mean, these are all unitary published account from a particular area, maybe Caucasian, right? As soon as you go and do the same study in India, in Mumbai versus say Patna versus Kashmir, they will be all different. So we do not have a very big understanding and of the, that diversity with age or whatever amongst the Indian population. No government is going to fund this kind of studies because they're a little bit expensive to do, and you cannot show direct benefit of knowing this, right? You can say that I will develop probiotics and postbiotics will be healthy for you. You will get money for that purpose, but just documenting the variability in in microbiota, I don't think you will get money for that. So that's something that we need to, as a policy, have a much larger, mission mode study to do. So, of course, so this is one of the things that we are trying to understand, how pathogen or how the microbiota interacts with an organism and how do they affect aging. So, in general, my lab has been very interested in studying the mechanisms of aging. And since my postdoc days, almost 20 years ago, I switched from plant sciences to animal sciences just to study the biology of aging. And and we study all kinds of things, right? So we are not just one lab focusing on one thing. We just, our interests are sort of goes from studying organelles like proteostasis in endoplasmic reticulum or mitochondrial stress to signaling and metabolism. Now we are interested in how the reproductive tissues age and how different parts of the body can control reproductive decisions. We are trying to develop some pharmacological intervention that can prevent or slow down aging. And we also interested in one of the diseases of aging, which is cancer biology. So if I have to tell you about aging, let me first, tell you about how I think about the process of aging. And, the first thing I ask myself is that, how do I define aging? If I have to study aging, and that was in 2003-4, when this field was upcoming. So this is not, not too much known about the molecular basis of aging. People were not thinking in one way or the other to define the problem. And so unless you define a problem, you cannot, address it. So what I thought is, why don't I see aging as a phenotype, right? It's a phenotype with which I can do genetics, just like Drosophila biologists do. Can we use the phenotype of aging and use it to understand the biology of aging? So that was, my crude way of thinking as a plant scientist. So, but it worked out pretty well. So how do you define aging? So the three ways you can define aging, and these are all very diffuse, right? It's not solid measurements. So an exponential increase in mortality rate with age, and this you can measure. And this measurement is absolutely conserved, whether you measure it in human, you measure it in mouse, C. elegans, Drosophila yeast, the lifespan curve is invariant. Although the number of years or days will have differ, but the curves are absolutely conserved. So this is a good measure. The other measures you have are physiological changes which happen with age. One of the great physiological measure is frailty. It's a very, very conserved way of measuring the progression of age. And also increased susceptibility to disease. That also you can measure by doing challenge assays. So you can define the process of aging by measuring one or two different parameters, although we absolutely love to do lifespan assays because that's an invariant measure. So not all organisms seem to age, right? You have seen these organisms, they show no signs of aging, but I can assure you their organ systems are aging and they will die of old age, right? So if anybody tells you that you don't look like you are aging, I think they are just scoring cookie points, okay? And so then don't believe them. Or they are your Facebook friends growing older together, and they don't see each other, very differently. I think that's how it is. Now, the important thing is, why do we study aging, right? So people have been trying to study aging for millennia. And one of the things that they wanted to study was, can we live forever, right? All kings and, everybody wants to live forever. And this is, yeah.
The parameters that you mentioned, I mean, it's not necessary that everybody will have the same parameter, same number of years of age, is it?
Yes, that's the only variant. So that
So-
x-axis will differ.
So somebody at 60 may look younger than somebody at 40, maybe. I don't know.
So those are special. I mean, those are flattery to me, because your biological age, as determined today by epigenetic clock, actually tells you how old you are. So it's not one parameter. You look. You can look younger, but your inside can be falling apart, and the vice versa. What happened? So
sense, you can plot and you'll see an exponential.
you see, yeah. So if you plot it, you will see an exponential increase in mortality over a period. So that also,
You
yeah.
have such a plot? I mean,
Huh?
just curiosity. You have such a.
Maybe I'll show you later, but not an exponential plot, but a lifespan graph. Then you can, being a mathematician, you can, just extrapolate it. Okay, so the reason why we study aging is not to live forever, right? So this has been there, in mythology. If you think about it, if you read Stephen Fry, for example, the gods don't die, and this has been sort of knit into the fabric of society, that there is a possibility that if you get the Holy Grail, for example, as you've seen in Indiana Jones and the Last Crusade, or if this beautiful painting from Lucas Cranach in the 1500s, which depicts the fountain of youth. So, as we biologists of aging think that there's a maximum age limit till the human body can handle, and that's around 121 years, because there's no scientifically documented evidence that anybody has lived past that. So that's the threshold. Now, if you think about how we are progressing as a society with bionics and all, I mean, like, you can live forever, probably. But it is not important to live forever. What I'm trying to get at is the length of life is not important, because you would end up having this Tithonus error. So Tithonus is a mythological figure, and Eos fell in love with Tithonus and asked Zeus for eternal life. And she forgot to ask about eternal youth. And Lord Tennyson has written this beautiful poetry about how Tithonus died slowly. I mean, he didn't die, but he became older and older, and then I think Eos turned him into a grasshopper or something, and that's how the mythology goes. So it is important that you live a long, healthy life. So that's where we are starting to change our parameters from lifespan to assays where you look at muscular efficiency with age, your your ability to focus, your ability to solve problems. So those are becoming better measures of aging rather than just the lifespan. Okay, so, and also the lifespan curves vary a lot. Your populations sometimes have a few trailers, right? So a few people live long, and and the rest of the population might have died out. On the other hand, some of the very healthy population lives for a long time and then just crashes. So all these kind of things happen. But the thing is that you should live a long, healthy life, and these kind of lives are available in nature. So this is a seminiferous organism. It reproduces only once. It frolics in the ocean for 8 to 9 years, and then goes back and spawns and lays eggs in the same place where it was born, right? So these are examples of extreme longevity, because they live a very happy, healthy life, and just when they have to die, they age rapidly over 1 or 2 weeks, and then die, right? So they don't wait to raise their children. But if you think that that's a selfish act, the seminiferous organisms die because they feed the next generation. Their carcass feeds the next generation of these fries. So in human beings also, there are, examples of extremely long-lived people living healthy lives, and these are, very great examples. So, if you look at people who are long-lived in the whole world, there are these blue zones, and this is a book written by Dan Buettner. He is a National Geographic explorer in residence, and what he found is that there are a few places, right from Okinawa to Sardinia to Loma Linda in California, where people, they have intrinsically long life. They live for 100 years. If you have read this book, Ikigai. So there, people are playing at the age of 110, right? Games every day, doing gardening. One thing that he figured out from their, analyzing what they are doing to live long is they are eating wisely. Again, bringing back that what you eat is how long you will live, right? So, I think this book is worth reading just for the sake of it. I mean, we wouldn't have worried so much about aging had aging not been linked to diseases, right? So as you grow older, your body tissue parts will fall apart, and your immunity, regenerative potential will go down, and you will be prone to more diseases of aging, and mostly they are metabolic in nature. And this is becoming an increasing problem in this world. I mean, this map I just put, it's like weird, because in 1950s, the average lifespan of Indians was 35 years. And within a century, it's 68 years. So now you can understand that you will, in 2050, 21% of the world population will be 60 years and older, right? So that is a humongous amount of, aged people in the society. Not only will that affect output in terms of work, but it will also kill a nation who have social welfare schemes, because the cost of maintaining older population in good health is enormous, right? So you'll end up having AIIMS as kirana shops in the city. So that's untenable, right? So in order to really understand the biology of aging and to prevent it, we need to know why we age, right? So the research on aging has materialized mostly in the last 20 years, I would say. Scientific, very hardcore, evidence-based research. And if you see that a review comes out every 10 years from this group led by Guido Krummer, and they map out all the evidences and say what are the hallmarks of aging. And this is a recent one. And very interestingly, these hallmarks grow, and the most important part is that not a single hallmark is refuted, right? So that they keep adding. So now you understand that the depth of research has become, come all the long way from alchemy during Lucas Cranach's painting to hardcore fundamental science in the biology of aging. So I'm going to give you some examples, but what I want you to understand is that there are some primary reasons for aging, and that is, genomic instability and blah, blah, blah. So these happen. And then there are some antagonistic. Antagonistic pleiotropy is a concept in developmental biology where some of the processes, biological processes, are meant for development. And they have actually no use when, the animals grow past development, and they become, counterproductive when they become adults. And there are certain examples. They are backed by evidence. And if you want to know, I can tell you a few of them. Now, finally, there's one integrative. All these damages, all these things that happen during the aging process also creates these kind of abnormalities like, altered communication, chronic inflammation, and I mark dysbiosis. Dysbiosis is one of the hallmarks of aging. So it has now become integrated into aging research. Now, quickly go through why we age. I'll give you 4 examples. If you are not biology students, don't worry about it, because this is not biology. You can understand, right? If you have seen children going through, the nursery classes with a tie, they keep chewing their tie, and by the when they go into class 1, half of it is gone. So this exactly happens in our chromosomes. So because when they divide, they keep losing the ends. Bad example. I thought about it that, if there are physics people, how do I explain the chromosomes? I don't have time. So this is it. So now when this happens a lot, the cells finally throw out the towel and say, I'm done. I either stopping to divide or I'm just going to die. Huh? Yeah, and allow, yes, they throw, actually they do, right? They, the fusion of DNA into the, yeah, so that way they kill themselves or they arrest or sneeze. They don't do anything, they don't grow, but they keep on putting signals that I'm sort of done with life, come grab me, and that creates basal level of inflammation. Okay, so that is counterproductive. Now, the other thing that does is that these are, mitochondria, the powerhouse of the cell. They make ATP, which is gasoline for the cells to grow. And when the mitochondria becomes older, they actually don't look like this. This is textbook picture, but they are beautiful, fibril-like structure. And when the cells grow older, they start fragmenting as if they have been hit with reactive oxygen species. And this is one a very good example that you can observe in the laboratory, that you give H2O2, they fragment, and if you compare them with an old cell, they are fragmented. In fact, this is very strange that cancer cells are also fragmented, but they go fine. So they use that fragmented, mitochondria to survive, and there's a lot of biology behind that. If you look at stem cells, which replenish many of your organ systems, these stem cells are exposed to a lot of abuses of nature, and they get DNA damage. So their DNA is damaged, and as a result, they get mutations. They change in DNA sequences, and if they gather too many mutations, there is a concept called double hit, and it goes into the mutations knock out tumor suppressors or tumor oncogenes in bad ways. They will form cancer, and if the cell detects it, the cell will say that you arrest and you senesce, you don't have to do anything else. And when the checkpoint is activated here, as I said, it will end up becoming a senescent cell. And this senescent cell, again, will throw out signals which will, elevate the basal inflammation, which is bad. Now, the last one is the amount of proteins that are made and degraded in the body has to be very equalized, and this is what falls apart with aging, and this is called problem of proteostasis. So all these things matter, but what we are trying to do is, instead of going for diseases of aging individually, Linda Partridge put this review together saying that why don't we intervene at the level of aging rather than individual disease, and try to fend off aging or delay aging and have a better health. And you will be amazed to know that there are scores of molecules which are in clinical trial trying to delay various processes of aging. And one of them you will already know, it's metformin, a drug that you use for diabetes. And the other one is resveratrol, which is the alkaloid in the red wine, right? So these are all in clinical trials. So we now shift a little bit to what I do. Otherwise, I'm not going to leave you giving a general talk, right? You have to hear what I do. So I use this wonderful worm called C. elegans. It's a beautiful nematode, and it has fetched a lot of Nobel Prizes also. Same organism with Sandhya works, and she's smiling at me. So it's really amazing organism, and you can ask all kinds of questions. Even if you are not a biologist, you can ask questions about biology and physiology and environment, everything with this model organism. And the best part is it is transparent. You can see through it under a light microscope. Very cheap to maintain. You can, we can do experiments. We do it in colleges. We go in schools where you can take your microscope and do fun experiments with the kids, and they get really excited. And this is backed by powerful genetics. That's our, tool. We have, AK-47s and all, just doing fantastic science with them. The most important thing is genome is conserved. So 70% of the genes that are there in C. elegans are also there in humans. So we can ask questions which are relevant to humans. And we are not doing anything in test tube, although we can grow the worms in test tube. Everything is in vivo, means that we are looking at real organismal level biology. Now, let me bring you back to the blue zone, because again, I want to remind you that how important your diet is. your diet is important not only to maintain the gut microbiota. I told you that what you eat determines how long you are going to live. Now, those who are trained in this area will know about signal transduction pathways, which are important in regulating various biological systems. And they would know that if you feed a lot, right, like, you feed pizza and pasta every day, you're going to go obese, and also these will in negative way affect this signal transduction pathway. On the other hand, if you restrict your diet and lower your calories, you will positively affect the same pathways. And I will give you some empirical examples of it, because molecular examples are boring. If you look at this very example, that if you eat more and you become obese, you will be prone to most of the diseases of aging. I've highlighted them, right? So those are the same diseases I highlighted when I said gut dysbiosis causes those diseases, right? So there is a general link between diet, microbiota, and diseases of aging. On the other hand, if you do an experiment, of 20 years, which is like 4 or 5 PhD life of yours, where you maintain these beautiful looking monkeys on low calorie or high calorie diet, you inevitably see that the ones which are maintained at low calorie diet live long and are healthy. And this tells us, and this is what Ullas had done for such a long time, and showed many different fantastic things. Now, when I talk about diet, I again remind you that diet consists of macronutrients and micronutrients, right? And the micronutrients are the ones which are mostly made by the microbes, like vitamin B12. So we got a little interested in this field some time back, and there is an interesting reason for looking at these things, because this is a little bit towards the side, but most of the human population or any animals, they don't get sick just by eating pizza for 7 days, right? Or having great health, they just don't start becoming younger. So what I want to tell you is that every organism has homeostatic mechanisms. Very important to understand that, right? So some people become sick easily, some people do not do, and we always, blindly ascribe it to their genes, right? So it is actually their genes that can become protective in one way, or they can make you prone to certain, disease of aging if you have those mutations. So what is known is people having this fat mass and obesity associated gene polymorphism, there are some changes in their DNA, they are more prone to become obese. But if you feed them Mediterranean diet, which people in Sardinia have, you can fend off this. They will, within a few weeks, their whole biological parameters change. And this Mediterranean diet is again something that promotes microbiota. On the other hand, if you have a mutation in the methionine reductase gene, I think I forgot the full name, methyl tetrahydrofolate reductase gene, which is involved in one carbon cycle, which requires vitamin B12 supplied by the microbiota, you start seeing defects in homocysteine metabolism, and these individuals are prone to cardiovascular disease. The only way to treat them, give them high amount of vitamin B12. So this tells you that how important the homeostatic mechanisms brought in by the host genes are important in maintaining a healthy life. Now, how do you study these homeostatic mechanisms? How do you study the interaction of a microbe with a host, right? If you just grow a host on a particular microbe which is not harmful, you will not see any changes. You need to have sensitized background. So in C. elegans, it is a bacteriovore. It eats bacteria for a living. And in laboratory, you can feed them different kind of E. coli bacteria, and E. coli do not make vitamin B12. They pick it up from the food that you're supplying to the media. Now, there are certain mutations, which I'm not going to go in details, cause these animals to become long-lived or short-lived when they're eating two different kind of bacteria. There are various reasons for that. But if these worms, for example, we are interested in this Richter, which I'm not going to talk about, but the FLR4 gene is a serine kinase. It can phosphorylate other genes. If you mutate this, if you have a gene which is kinase dead, it doesn't have that catalytic function, they start living long on this H bacteria and not on the OP50. I'll show you this as a lifespan data, which I promised I will show you. So this shows that these animals, the mutant animals, when fed HT115 diet, are long-lived, much longer than their wild type. On the same mutant that are grown on the OP50 bacteria. And Sonia, who is now a faculty in CDRI Lucknow, she painstakingly found out why this is happening. And what they found is that when this kinase is mutated, it activates a serine kinase pathway, which is an innate immune pathway, right? So the pathway which gives protection against against microbes. And this in turn activates many cytoprotective genes which protect against aging. And as a result, these worms start living longer and having stress tolerance only when they're fed high B12 diet. So again, I mean, doing this kind of assay, you would not like to do them in the lab. So you need to find alternatives, which we found. And all we did is we took this gene, tagged it with which is GFP, which now fluoresces based on the levels of the levels of the activation of this pathway. So if you see very easily that when this mutant is fed with OP50, it doesn't glow, but when it's fed with HT115, they start glowing. So this is our base assay. When I say you that it glows, you know that the lifespan is increased, the stress tolerance is increased. So I will not show you any more lifespan data. So next, when after Sonia, Tripti started asking what is so different between these two microbes, the same E. coli, right? It's two different strains, but what is different? So did a lot of experiments, 1 year, couldn't find anything. Then it struck her that the only difference between these is the level of B12 that they give to the worms, right? So she found that the HT115 bacteria is very rich in vitamin B12 and OP50 is deficient in it. And if you supply OP50 with vitamin B12, it does the same thing. The worms now start living longer and a healthier life. So this was her whole PhD work, but that was not all. She did a lot of metabolomics assays and genetics to find that there are two pathways which are important in this phenotype. One is a metabolic pathway, and the other one is a signal transduction pathway. Metabolic pathways produce metabolites which are important for biological processes, and the signaling pathways signal from one place to another to activate genes which are required for certain biological phenomenon here, which is to have a long lifespan. Now, this pathway is very interesting because this pathway requires B12, which only the bacteria can supply, right? So here, again, I tell you that these are interspecies interactions, and the bacteria is actually providing metabolites which control host longevity, right? So now this is what we had when Shabnam joined my lab. She asked, are these things happening in the same cell? Because, as biologists, you do not have everything happening in same cell. It can happen all over the place. You need to find that out. So she started her PhD asking that, can I find out where this metabolic pathway is occurring and where is this signaling occurring and how they integrate with each other. So what did she do? And this is very easy in C. elegans research. You don't need to do any cutting edge, anything. It's just putting things back where they belong. So first thing is that you have to take things out of C. elegans, and that you can make by making mutations. So you take the mutant and you kill the 1-carbon cycle. You kill it all over the body, right? And then you use genetics to put them back in different organs. So she put them back in the neuron, she put them back in the intestine. And what did she find? She finds that when she puts them back only in the neurons, you start seeing the glowing worms, meaning that the pathway is complete, the gene is now expressing, and this is responsive to B12. But if you put it back in the intestine, it doesn't matter. So that tells that metabolic pathway is situated in the neurons. Now you go and ask. Yes. Okay. You want?
They're that well behaved, these worms?
No, they're not well behaved. We just anesthetize them.
And what does it mean that there is more of them that is green versus less?
Yeah. So this is, if you sorry, this is the
So
other.
is it, I mean,
the-
each line is a worm, right? And
Oh, sorry. It's
some fraction of it is
Hold on, hold
green
on.
and not green.
Yeah, this is not the slide I showed. This is a slide I showed. Okay. So these are worms lined up. This is their lower gut region where this specific gene, this is innate immune gene, it expresses. And this is quantified here. So if you now put, so this is the double mutant, which doesn't have glow. You put only in the pan-neuronal, use the pan-neuronal promoter, and we rescue the GFP and rescue the lifespan stress tolerance. You put it in the intestine, it doesn't affect that much. So that's how we. So next, we asked where is the signal transduction pathway working? So we showed the metabolic pathway. Now we asked where the signal transduction pathway. And we did the experiment a little bit differently. So what we did is we took the endogenous gene and used siRNA to knock down the expression. Okay, so sorry. So when you knock down the expression and you see that only when you knock it down in the intestine, you get rid of the GFP. You don't do that in the neuron. Okay, so that means that the signal transduction pathway is working in the intestine. So now you have these two things working in two different regions of the worm. One in the worm neurons, the other one in the gut. And the next question is that how do they link these two? So when in C. elegans, when you don't know anything, you just do screens. Okay, so we did screens, and the screen is very simple. You take these look like brains, but there are neurons. Okay, and connecting they look like bones, right? And here they're not bones. So they are neurons connecting the presynaptic and postsynaptic neurons, and they communicate with either neurotransmitters and in some cases neuropeptides. And so what we do, and this is what we have learned because we don't know neurons, we learned from Sandhya and Kavita and Anindya that you need to knock down certain genes to claim that they are functional, their function is in neurotransmitter release or neuropeptide release. And to our surprise, that when we knock down both neurotransmitter and peptide release genes, we got suppression in that GFP, right? So that told us that both these neurotransmitters and neuropeptides are required. So that was fascinating for us. So we went through all the neurotransmitters that are made by C. elegans. We knocked them down by RNAi, and we found that involvement of something very interesting, very close to Vidita's heart, is involved. And so we followed that. We also got dopamine. We don't know why dopamine is required. We're working on that. I have no comments to make on that. So now what we do is you need to make reagents. So what we did is we completely knocked out serotonergic signaling in C. elegans by knocking down this gene, which is the sort of rate limiting step in this process. And you see that the GFP is completely gone. Now, very beautifully, if you give C. elegans serotonin, if you feed serotonin to C. elegans, you can get back this gene expression, right? And you get back lifespan, you get back osmotolerance. And that's what we were, sort of discussing, that how C. elegans can pick up neurotransmitter right from the food. That's fascinating to think of. So now we know that this is a serotonergic neurotransmission is, required. And for a biologist like me, I have all these problems because I don't understand that much.
Question, maybe we can do it after they finish, but it will be interesting to know how serotonin passes through, in the sense that how is it uptaken?
Exactly, that's the kind of experiment
that membrane associated also. So yeah, anyway.
So nobody has done that very silly experiment to block the reuptake inhibitor and give serotonin and ask whether can it still work. Yeah, that is
sure.
We just got the mutant. We'll do that experiment.
Okay.
So, as I was telling that, for me, things are not obvious, right? So if I look at the graph and somebody tells me that serotonergic signaling is involved, I think that, this can be downstream of FLR4, can be here. How do we figure that out, right? It's not obvious to me. So what we did is we did some supplementation genetics, very easy experiments. You take the mutant and you also knock out the neuronal metabolism of one carbon. And then you ask, if you block it there, can you add serotonin and get it back? And when we did that, we found that even blocking, taking out the upstream activators, you can just put the downstream stimulator, and you get back gene expression, get back lifespan. So it tells us that it is definitely downstream of 1-carbon metabolism. Now, we also did experiments. I think I haven't shown it for the interest of time. We figured out which neurons exactly, it is involved. So we figured out by putting back in different serotonergic neuron, we identified that this is ADF neuron mediated, right? And also this response to B12. At every step, I had to ensure that it is responsive to B12 because that's the interspecies interaction, right? So having done that, we looked at what is the receptor, and we found that this receptor is at ion channel gated receptor in C. elegans, and that doesn't express in the gut, which would have made our life so much easier because it would complete the whole circuitry. It expresses only in certain interneurons, right? So we sort of had a model where we know that changes in B12 dependent metabolism fires serotonergic signaling, binds to MOD1 receptor in the interneuron, but we still had a gap. So we looked at literature, did a lot of dirty experiments, and we figured that this could be because of the neuropeptide, because that, you remember, that came up in the neuropeptide came up also as a downstream effector. And through a lot of literature search and some very intelligent thinking by the PhD student, we homed in on a glycoprotein hormone FLR2, and we showed that if you knock down FLR2, you get rid of the GFP, and that cannot respond to serotonin anymore. So possibly this FLR2 is downstream of this signaling. Now, I mean, this could be anywhere, right? So the only way to prove it, if we can drive this neuropeptide in the MOD1 containing interneurons, right? So then only we can prove that this neuropeptide is being secreted by the MOD1 containing interneuron. So that's what she did. She drove that neuropeptide into the MOD1 containing interneuron, and that brought back lifespan, that brought back gene expression, brought back stress resistance. So she also figured out what is the downstream receptor on the intestine, because somebody has to take the FLR2 neuropeptide. And then she completes the story by showing that this binding leads to phase transition of this TOR interleukin-like domain containing protein, which sits upstream of this signal transduction pathway and activates it. So this is the whole story that I wanted to tell you. But in the last 1 minute, I'm going to tell you how this circuit not only affects behavior, not only that micronutrient from another species affects longevity, it affects behavior also. And we can do these cool behavioral studies where we just ask the worms to choose between two food. We grow them on condition them either on high B12 or low B12 diet, and then ask them to choose between this high B12 food and low B12 food. And to our amazement, we found a complicated looking graph. But let me make it simple because it's very interesting. As I was telling, that wild type worms really don't care what they're eating. They don't choose. They just go eat whatever they get. As soon as you have a mutant of FLR4 right here, yes, you get a choice for only high B12 diet. They all move towards high B12 diet. If you knock out the metabolic pathway, it comes down here, right? You put it in the neuron, you rescue it. You put it in the intestine, you do not rescue. You put it in all the serotonergic neuron, you rescue. You put it only in the 2 ADF neuron, you rescue behavior. You rescue lifespan, you rescue the behavior, right? So this is a gut-mediated behavior. Why? Because now you look at how these worms without the signaling pathway in the neuron, in the intestine. We put the signaling pathway back in the neuron, nothing happens. When we put the signaling pathway only in the gut, it starts choosing the high B12 diet. And this, I end by saying that, there's a saying that your gut feeling, right? Your gut determines many things in your life. This is one of the coolest examples that we have, that a signal transduction pathway emanating out of the gut tells the worms which food is good for its health, which food will give it benefit of health. So with this, I end, and this is my lab. This is actually 70% of my lab. And I was telling, my lab has a lot of people. Actually, very interestingly, I didn't know this guy. he was standing here taking photographs. Then I realized that he's a new recruit. He works in AIIMS, and he helps us, collect samples from patients and brings it to the laboratory. So, so he's a new guy. A lot of trainees work in my lab, and I have students with Rajesh Gokhale who works on microbiome, pathogenic organism and C. elegans interactions and how that is evolutionarily conserved. I have people who work specifically, two PhD students working on cancer biology, and lot of trainees. So with that, thank you. I hope I have kept time. No, I
Stick.
went over time by 10 minutes.
No. Don't worry about it. That's fine. Time was fine. So, questions? Yes.
Okay, so maybe if I take the example of like B12, which basically is influencing metabolism and so on. So these experiments, when you do, is it more like a trial and error kind of things, or there is a reason for choosing B12 or not something else?
Good, very interesting point. So the student, so see, there was no reason to immediately, think that the two bacteria will be different by B12, right? So the first 1 year of her PhD went looking for LPS structure. So because there was a publication which showed that LPS structure determines how long a particular mutant will live. So we did all kinds of bacterial work with different LPS length and all, couldn't figure out. So what came into what actually changed the dynamics, that when you do not, you don't have any clue, you do RNA-seq, and we are very good at doing that. So when we looked at RNA-seq data, there was telltale signs that one carbon cycle was involved, and the only way one carbon cycle can be involved is if there is a B12 input into that. So we looked at folate, we looked at other vitamins in that. Folate also plays the same role. It's just to increase the flux into the one carbon cycle. Folate you can get from diet also, and also from the microbiota. But we followed vitamin B12 because that was more relevant to us. And when we measured vitamin B12 in the bacteria, in the worms, and stopped, vitamin B12 uptake in the bacteria with bacterial mutations, all kind of stuff we did. Yeah.
Hi. Not from the field, very nice question. That is, so in case of mammals where you have the offspring usually takes a longer nursing time, how does these concept of antagonistic pleiotropism are still relevant in these cases?
Can you little bit elaborate on the question?
Yeah, so basically, so there are concepts like this grandmother hypothesis and stuff like that, where it demands the longevity of female even after menopause. So like that, but in case of this pleiotropy conditions, so it says that the longer longevity or something is not essentially good for the species.
So antagonistic pleiotropy, I mean, the way I would explain it to you, and as I understand it, is a process which is important in development. For example, mitochondrial dynamics, because when you are growing from a single cell into an whole organism, the process of development requires a lot of mitochondrial input in terms of energy, right? So that is absolutely important for the development part of the growth. Now, what we also know is if you now take these worms, which are, if you now think about mitochondria later in life, probably the mitochondrial metabolism changes in a different way because it is not that important in the biological processes when that organism has become adult. It's not that extensively used. And there are evidences which show that if you knock down the function of mitochondria in adults, you can actually extend lifespan. So that tells us that while mitochondria, if you knock out mitochondria, when you knock down mitochondrial function during development, you will get arrest, you will get larval lethality. But if you knock down later, when it is not that extensively used for development or cell division or whatever function, you can actually have beneficial role because you're toning down the mitochondria. When you tone down the mitochondria, it's not going to have as extensive electron transport. It's not going to make as extensive ROS as it makes during aging. And that's why you see the benefit. So that's a, sort of theory of antagonistic pleiotrophy. That's what I
So
understand.
is it very species dependent or something like that?
It should not, right? Because I mean, the whole point is that we cannot do these experiments in mammals to that way because C. elegans or Drosophila is very easy, and both these organisms you have seen that this is how it can work. But in mammals also, where will you disturb the function, right? That's also, are you going to disturb the mitochondrial biology in the muscle? Then you have to look at muscle specific function. Not organismal wide phenotype can be seen that easily in mammals. That's probably the difficulty why we don't clearly understand the boundaries there.
Thank you.
So quick question. If you do an S-adenosyl methionine supplementation, is that as effective as vitamin B12 in your 1-carbon cycle? And can you just feed your worms SAM and then see any?
Yes, we did all those experiments. So you change the flux and it works.
Okay,
You
so
can get folate.
so anything in that cycle
yes,
would
yes,
be.
yes,
So the other question was for serotonin. This is a neuronal release of serotonin.
we did the gut.
Yeah.
cells. We did that experiment.
And there's no
No,
gut
no,
serotonin
no,
that's
no,
playing a role.
No,
So that's sort of quasi surprising, only because there's recent evidence now that there's serotonergic production from bacteria, but also otherwise in
right.
enterochromaffin cells, etc. Other cells. You think this is unique in the context of worm, that it's a neuronal signaling pathway that's driving serotonin, and that in other species, gut serotonin may be playing the same role of potentially influencing? Or what are your thoughts on this, given that there's more serotonin in the gut than there is?
There is,
from the
yeah.
nervous system?
So what we did, we took a gut serotonin, PALM1 mutant, and that didn't show any effect, right? So that means that the neuronal serotonin is sufficient to do all this. So we haven't really done. How will you do it? Yeah, we have to think about it. I mean, why? But that was just a simple experiment that this didn't work, so that is sufficient. We didn't look at the other one. So that's about it. But
Maybe just there are a few questions. Arnab? Arnab, that's it.
Hi. So sorry, this is a physicist asking a very naive question. So I guess my watch has no clue of how much of my DNA is chewed up or the ends of the tire chewed up or whatever, but still it tends to massage my ego by telling me my fitness age, right? Which it seems to calculate by my resting heart rate, VO2 max, BMI, whatever. Is it actually possible to estimate longevity or age or whatever by external parameters, not knowing how much of the DNA is chewed up?
okay.
Or is it just like, massaging my ego by telling me that, hey, you're doing fine?
No, I think if you what you mean by the external parameters actually are clinical parameters for measuring how healthy you are. Right? How that is exactly what you can measure. It is not a great measure or predictor of your lifespan. You can predict that if you are, exercising, if you are a runner, for example, you would probably, under the best of the condition, live long. But it is not probably a great predictor of your longevity. That's a different tool. So the tools that are available to actually predict longevity are based on internal parameters more than external parameters less. Because you see the problem is, if you think about metabolic homeostasis, it differs so much between individuals. A very thin, frail-looking guy need not be obese to develop cardiovascular diseases because their threshold of fat storage is much lower, right? On the other hand, a very big man or woman can absorb a lot of these metabolic changes and still be healthy. So there is this parameter called healthy obese, although, there's a clinical parameter, healthy obese. And when you do clinical trials, you actually categorize them based on that. So I think that external parameters, it's not great predictor. So yeah. That is, I think, part of this, is it great predictor for tissue intrinsic aging. But again, at an organismal level, there is only one great paper from long time ago, which showed that it is causally linked to aging, right? So if you look at that paper, they had to bring all the tumor suppressors alive and then overexpress telomeres and show that if you overexpress telomeres and you maintain the telomeres length, the animals actually live longer, but do not develop cancer, because otherwise they will develop cancer and die when we cannot decouple these two. Yeah.
I'll ask very quickly. So you flashed one slide where you were showing different parts of the world with average lifespan, higher average lifespan. Japan was there, that is understood. But what about California? If I remember, California was also there.
Yeah, it's Loma Linda. Yeah, I have never been there, but this is in the book. So he has identified several areas where the average lifespan of a population. So this is also not California as a whole. It is not the whole of Japan. It's only the Okinawa prefecture, where there are few villages where they are extremely long-lived people. So even I don't know how, because I read that book, Ikigai, I know about the Okinawa prefecture and that, but I don't know how large or small this Loma Linda population is, but we can look it up.
So you just said something extremely intriguing, that telomere length predicts an individual organ's age.
Yeah, because if it is a stem cell aging, for example.
So do organs not age in sync? Surely somebody must have done heart, liver, kidney,
it doesn't age in sync.
Oh, so you may have a really young kidney and a old.
Yeah, it's possible
liver.
because, see, depending on the abuse on that particular organ, that amount of reactive oxygen species, that organ will. If you are a heavy drinker, do you think that your liver will stay young because you are just young?
No,
I don't.
So not real world situations in mice in cages. Do organs not age in sync? I'm just.
Can you? So if you look at, how will you measure that? In the sense that you can measure, say, the aging in liver and compare them between liver. And you are saying that you measure the telomere length in one organ versus other and say whether they're interesting. The worms, they age differently. That we have known. But what she is saying is that provided the worms, that organs have stem cells and that stem cell is rapidly dividing, going by the the size of the telomere, can you predict? I would say that if you see the number of senescent cells in that organ, that would be a better predictor, because the telomere length will differ at what stage of cell division each of this organ is, right? So I think that way, if you look at, it's a good measure, right? You look at the number of senescent cells and that you can stain, and that probably will give you a better idea. I'll read up more on this interesting idea of yours, and I'll get back to you.
Thank you, Arnab. This was fascinating discussion, but we have to stop the colloquium at some point. And
I'm here whole day tomorrow.
Yes, and Arnab is actually here for the next 2 days, 15th and 16th. So.
Yeah, don't.
And we are inviting all of you for some coffee and snacks, very healthy snacks like chips. So yeah.
Thank you, sir. Thank you. Thank you.
Clarify that the tie idea did not come from child.
No, you didn't.
Yeah, this has to be clarified by her immediately.
Yeah, thanks for that talk. I enjoyed it very much.
Thank you.
But let me just make sure I understood something you said towards the end about.
Can you
behavior.
switch it off, or they will help?
About behavior, which you spoke about at the end. So as I understood it, I may have misunderstood what you were saying, that the sort of, the worms which you had created, which basically genetically were modified to require B12.
Yes.
To sort of, where B12 had an effect on their longevity.
Yes.
Those mutants actually preferred eating B12.
B12, yes.
That sounds like magic to me.
That's magic, even to us.
Why on earth would it actually sort of work in that way?
So, yeah, I mean, because that benefits that